All right, let's talk about immunity. This is the wrap-up video. All right, adaptive immunity, it breaks up into two. So the easiest way to understand it is that it all starts in the bone marrow and ends up either being a humoral immunity or cellular immunity. But let's figure out how that works. So it starts in the bone marrow, and we'll talk about cellular immunity first. So everything is really produced in the thymus gland for cellular immunity, and it produces T cells. And then what happens with the T cells, we go down here, they actually clone into cytotoxic T cells. So a foreign antigen becomes introduced. These T cells respond and say, oh, this is not supposed to be here. Then they clone and become cytotoxic, which we'll talk about a little bit later. That's known as cellular immunity. And things that are activated from cellular immunity are viruses, fungi, and cancer. Those are the main things. So bone marrow, then these derive from the stem cells. So whereas the cellular immunity is primarily in the thymus gland, the humoral immunity takes place more in the stem cells from the bone marrow, which then turn into lymphocytes. And from the lymphocytes, there's B cells. So when a foreign antigen or a bacteria, which I have here starred, is produced, it produces antibodies. So that's why we divide it up between humoral immunity and cellular immunity. So when you think cellular immunity, you're thinking viruses, fungi, and cancer. And humoral immunity, we're thinking more bacteria and antibodies. Again, cellular immunity, viruses, fungi, cancer, we're dealing with T cells. And humoral immunity, we're dealing with antibody production, and that's to kill bacteria. And all of those, when we get to the T cells and B cells, they work together with each other to help each other out. Where do we find these? In lymph, in the spleen, and blood, mucosal tissue, lymph nodes. So going back to the humoral immunity with antibodies, antibodies are basically called glycoproteins. And those are divided up, and I just put the first three common ones, IgG, IgM, and IgE. There's also IgA and IgD. IgA is in body secretions. IgD, there's really not too much known. But these are the three most common that you're going to see as you practice as a nurse, and even go further. So IgG is, actually I'll just keep the screen. IgG, 85% of it protects against infection. So what IgG does is build up antibodies, just like discussed in the past here. So an example of IgG would be, so because it protects against infection, it's more or less if you've been exposed to something, and then it builds up antibodies in your system. So let's just say you had chickenpox as a child, and then you're an adult, you become exposed to the chickenpox, you already have antibodies built up against that. Now, you could also go on to develop shingles, but again, most people, I'm not going to give a percentage, but many people have enough antibodies built up that you will not even get shingles, but you should not get the chickenpox. And that all has to do with IgG. If I have somebody that's had mononucleosis a couple years ago, and they come in with symptoms again, their IgG may be elevated. That tells me they've had past exposure. IgM is the first thing to go up. So that tells me that they have more of an active infection. And you see how these things work together, viruses and bacteria, because if I look at the Epstein-Barr virus, mono, I still draw, and I can tell if the antibody, if it's an active infection now, IgM, or if they've had past exposure, IgG. All right, so let's work off and talk about viral infections. I put a whole bunch of terms on here because this is basically a virus in a nutshell. They are small, simple organisms, and some of them are enclosed in a lipoprotein envelope. There's also some that have RNA, some have DNA, some are double strands. So all of them are maybe just a little bit different. Also, they mutate. So they're unrecognizable by our own immune cells. They also can cause malignancy within the cell. And they also replicate. They attach to the receptors on the host cell, which I made a little cell down here, and enter into the cell. So when a virus, what happens is it attaches to the cell first, then it penetrates, releases this nucleic acid, and then replicates. So then what happens, they start assembling and releasing new viruses out into the system. So why do viruses survive so much? Remember from your slides, they replicate very fast, they survive inside the cells, and then they suppress the immune system, so that doesn't help your body to fight it off. And it also has an antigenic variation, so they can be, characteristically, they can be a little bit different. And what is actually scary about viruses is that they can mutate, like I had said here, and they can make new strains. So, and to talk pathophysiology terms, they're surface proteins that can shift their antigens and make new and virulent strains. And so this goes back to the flu. Let's put influenza, so we don't get that mixed up from GI flu, which is what we've known for years as the flu to be, but it's not. This is how they determine what strains to put into the new flu shot, because every shift, antigenic shift, which is about once a year, they look back and say, okay, what do we expect the new viruses are going to be within the next year? So H1N1, or variation of H1N5, there's a whole bunch of them. And now we have this new coronavirus out, and there's really no cure for viruses. And so that's what makes it so difficult. And that's what makes it why the CDC put out some precautions against this, because we don't want a lot of this coming in our country. We want to stop it. We don't need a coronavirus epidemic here. We already have an influenza issue. So quickly to talk about bacteria, which is made of a chromosome plus DNA. There are exotoxins and endotoxins. So bacteria can be gram-negative, gram-positive, which I'm not going to get into detail with that. But there's bacterial toxins, exoproteins and enzymes called neurotoxins, and that's the exotoxins. Endotoxins are from the gram-negative walls. So basically toxins cause excess inflammation. But how does bacteria overcome host defenses or the immune response? Well, they can biofilm or put a capsule around itself, which is encapsulation. They have either biofilm or encapsulation, so don't forget that. They also can mutate and produce slime, as well as some enzyme production. So I put the most important ones that you should really try to look up and understand as how they overcome the host defenses or the immune responses. Make sure you look up the difference between innate versus acquired immunity. But one thing to know about innate immunity is that it's fast and the barrier is really mechanical or chemical. Innate immunity also uses the complement system, natural killer cells. So just make sure you review that. All right, so to dig deeper, what exactly is phagocytosis again? It's opsonization of a microbe, it's engulfment, fusion, destruction, and release. So neutrophils come first, and then destruction happens, and then it produces pus. That's what you got coming out of those yucky abscesses, infections. All right, so let's talk quick about hypersensitivity reactions. We have type 1, 2, 3, and 4. I'm going to just hit on these really quick, but type 1 is immediate. So when you think type 1, you're thinking peanut allergies, bee stings, pollen, also just environmental allergies, cat dander, dog dander, those types of things. So an antigen produces mast cells, and then that releases IgE and FC receptors, and so FC receptors are on the mast cells. So when we think type 1, we think mast cells, we think IgE and the FC receptors. And what happens is that releases histamine. So we have H1 and H2 histamine response. So that's why we take antihistamines if we have environmental allergies. And H1 blocker is when you think of Benadryl or Allegra Claritin. H2 is when you think of ranitidine, which would be your Zantac, or famotidine, that's Pepcid. There's a whole bunch of them over the counter. But we also have H2 receptors in the stomach, which is why we take Pepcid to help block the H2 receptors that's causing inflammation in the stomach. But what ends up happening, what's dangerous about it, especially if you get a bee sting or peanut allergy and you eat peanuts, it causes vasodilation and bronchospasm and edema. So your vessels dilate, you spasm in your bronchial area, and you swell. Your throat swells up because of all of this histamine release. Usually, well, not usually, let's just say genetics. If you have a genetic predisposition, which there is many people who have that genetic predisposition, you usually do produce more IgE, and you usually do have more Fc receptors on the mast cells. But just understand what a type 1 is. A type 2 hypersensitivity is more complement activated, or in other words, mediated by antibodies. Also, IgG and IgM, and they cause cell destruction. So, for example, would be hemolytic anemia, blood transfusion reactions. Those are the most common types of a type 2 hypersensitivity. We don't see it very much. When we think of type 3, it's again, antigen antibody immune complexes in the circulation that ends up just injuring your vessels and tissue. So autoimmune diseases, lupus is one of them. Whereas type 4 is more of a delayed response. So if you get a TB test, which showed in the slides, you end up getting a delayed hypersensitivity response. Another example of type 3 would be glomerulonephritis. Because what happens is these immune complexes gets into the circulation, and remember, it starts injuring the vessels and the tissue. Think about autoimmune disorders. There's still a lot of research out there regarding how people end up getting these autoimmune diseases. We do know that there's genetics involved in this, a change in gene expression. But usually what we find out that they need something to trigger the event. So let's take multiple sclerosis, for example. How do you get multiple sclerosis? Sometimes we just don't know. Perhaps someone had a genetic predisposition. I'll use an example of my sister has Graves' disease and MS, and her daughter has celiac, and my grandpa had type 1 diabetes. Well, that all lies along the same gene, or HLA alleles, I should say, to make it more exact. So what causes it? Environments. There's a lot of theories out there that MS is from you get a viral infection. So if there's a combination, let's say, of a viral infection, let's say you get metal fillings, which is another theory. We do have some really good research out there that it's hereditary. So if all those things that maybe can trigger the event for that gene expression to be triggered and end up starting with symptoms and consequently MS.